Next up, we have our next session will be on diffuse intrinsic pontine glioma, DIPG, otherwise known as DMG. We have another fantastic lineup. It's going to be moderated by Dr. Zacharoulis, and we'll get started. We have a couple people joining online as well.
Good morning. I would like also to thank the foundation and in particular Dr. Polovic for putting all this together for us. This is about a pediatric brain tumor called diffuse midline glioma. And my name is Sergio Zacharoulis. I'm a pediatric oncologist, and of disclosure, I also lead the drug development in Bristol Myers Squibb in addition to my Columbia appointment. So this session is about diffuse midline glioma and how we capitalize the newest technology with focused ultrasound in advancing the treatment. First slide, please. I see here we welcome our panelists, which I will introduce during the presentation. I'm going to give an overview of diffuse midline gliomas and the focused ultrasound. And as I speak, I will introduce each one of them. If we can start with the presentation. So is there? I cannot see them. Okay, let me. So. If we go back to the beginning, this is not the first slide. Yeah, next slide. Yeah, perfect, thank you. So diffuse midline glioma is a lethal pediatric brain tumor. It's probably the most devastating human cancer, primarily because it affects children. It's a brain tumor where children become progressively neurologically devastated, and unfortunately, the very vast majority, practically all of them, will die on average of 1 year. The past decade, we have made tremendous advances in understanding the molecular biology of diffuse midline gliomas. We discovered new mutations as the H3K27, which changed the way that we call the tumors. We have performed hundreds of clinical trials, including chemotherapy, biological agents, immunotherapy, vaccines, and unfortunately, as it stands, the only standard of care remains radiation therapy. Radiation therapy provides only a marginal benefit of approximately 3 to 4 months. So there is a tremendous unmet need for this disease. Now, there is a lot of work that is being done from various groups on focused ultrasound. Next slide, please. Primarily to address one of the barriers that we have in advancing the treatment, which is, in addition to the tumor heterogeneity, is and of course, the drug resistance, is also the blood-brain barrier issue, which we are going to describe how we address it later on. Next slide. Thank you. So, I don't need to state this to this audience here, but the blood-brain barrier is a very dynamic functional unit, which plays a major role in neuro-oncology. What is the evidence that we have for blood-brain barrier being an issue for diffuse intrinsic pontine gliomas? There are several aspects. First of all, pontine gliomas or midline gliomas typically are not contrast enhancing. This, however, is a very weak evidence, as we know, primarily because the issue of contrast enhancement does not necessarily mean much about the blood-brain barrier, but at least is weakly supportive of the notion that the blood-brain barrier is intact. Also, there is more of an aspect around the anatomical location of the midline gliomas, especially in the pons, where it is clear that the brainstem has been shown in preclinical studies in animal models, that it is not as permeable as the supraendothelial compartment. The other issue is that it has been shown that the capillaries, also the density is less, which makes it even harder when the tumor forms. And we have more evidence of that based on some autopsy data. So there is a high suggestion based on that the blood-brain barrier is an issue in diffuse pontine gliomas. Next slide, please. So I think this was mentioned earlier. So in order to address this, there are several technologies currently existing that are addressing the pontine gliomas. The main ones that are relevant to the clinical trials that we're discussing are one of them is the Incytec, which already has been mentioned, with the advantage of being non-invasive, covering a large portion of the tumor, but requires an MRI and sometimes probably anesthesia for little kids. There is the invasive aspect of excuse me, the French group has the most experience with, where having the disadvantage of being invasive procedure. And then there is the Columbia device with the TheraWave, which is non-invasive, it's mobile, but has the disadvantage of covering smaller area, at least at this stage, similar to Navifast. Next slide, please. So what do we know about the preclinical work that has been done in diffuse midline gliomas? There are very few groups throughout the world that have been working on it. And I will just summarize with this slide the high-level data. So primarily, we know that even in models with diffuse pontine glioma, using focused ultrasound is safe. It doesn't lead to any death or severe toxicity due to hemorrhage. And when we look at the behavioral and histological and physiological changes within the animal models, these remain intact. Also, it is quite repetitive. It is seen that, quite consistent, that the blood-brain barrier opens and then reversibly closes roughly within maximum 72 hours after the sonication. The main agents that have been tested are primarily chemotherapies such as doxorubicin from the Canadian group, temozolomide from the group in Netherlands, and as well as epigenetic modifiers like panobinostat from our group at Columbia, as well as etoposide. Now, in terms of the data that we have, the Canadian group has shown that doxorubicin and other groups as well, the intratumoral concentration can increase fourfold by the using of sonication, and the etoposide even fivefold within the tumor. It has to be noted, though, that mainly in these preclinical studies, there was no clear survival benefit in the animal models. Next slide, please. So there are a lot of questions looking at the overall, the preclinical work that has been done for diffuse midline glioma that are a lot of data that have not been explored yet. For example, is the lack of survival benefit that is being observed now based on the inappropriate use of models? How do we address the tumor dissemination? For example, we know that when we use the focused ultrasound to open the blood-brain barrier, there is evidence of increased circulation of cell-free DNA. And how does this impact the potential of metastasis? Or is it because the disease is so lethal that we don't even have time to see the development of metastasis? How does the opening of the ultrasound affect the immune environment within the bones or the midline? How do I address the tumor heterogeneity despite the use of the ultrasound? What are the proper pharmacokinetic and pharmacodynamic studies that we need to do to ensure that the concentration of the drug that it is at therapeutic level with a biologically effective dose remains constant enough to provide its effect? And what is the gold standard for the evaluation of the blood-brain barrier opening? Next slide, please. So coming to the clinical studies, we will hear from the panelists. We have with us Dr. Beccaria from France, who is also in the panel, who is going to describe us the update on the status of the study using carboplatin, using the SONOKID phase 1 study. I will clarify, I think this is for supratentorial tumors, but we will hear more about this. Next slide, please. Also to introduce my esteemed colleague, Dr. Packer, who is honoring with his presence here. He is obviously a pioneer in neuro-oncology and pediatrics for decades, and his team has developed 2 studies. In fact, one with low intensity focused ultrasound with the use of doxorubicin in collaboration with the team in Toronto. And the other more of a sonodynamic approach with 5-LA for pontine gliomas. And Dr. Packer will give us a brief update of the status of this study. Next slide, please. Dr. Javad Nazarian, who is also in the panel, has been leading the translational efforts in focused ultrasound and midline gliomas, and he has created an excellent network with Dr. Wu, Dr. Viljasevic, and Dr. Manson in order to address the changes in the immune system and also overall the molecular biology of the pons with use of focused ultrasound. Next slide, please. And the team at Columbia, next slide, please. He's done with Dr. Wu, Dr. Zeltoni, who is also here at the panel, have done a lot of work, both at the preclinical and clinical level, evaluating etoposide and also panobinostat at the preclinical level. We had at Columbia the first clinical study with diffuse midline glioma using Dr. Konofagos' system with a mobile, non-invasive, without the use of sedation for the first 3 patients. And we will hear a little bit more about this by Dr. Zaltoni, as well as the future plans. Next slide, please. And Dr. Dennis Varvoorden, who is also kindly joined us from the Netherlands virtually, who is also a pioneer in the field with drug delivery in pontine glioma, and he will mention his update with use of temozolomide in diffuse midline gliomas. Next slide, please. So there are a lot of issues that remain controversial in terms of, or probably unanswered. For example, what is the appropriate trial design when we deal with both a device, new drug delivery method, and a drug that potentially is even novel as well? How can we move on from the recommended phase 2 dose-defining approach, since there are so many parameters to take into consideration when we use the focused ultrasound? What is the risk of dissemination by using this approach? And also, do we treat patients who have distant disease, what is the technology of choice, and what is the way to standardize how we monitor the disease. And I will close that, and we'll start with the questions to the panelists. Thank you. So, as a first question, perhaps maybe I'll go around the panel. I will ask Dr. Parker to just briefly mention the current state of the proposed studies, and then we'll go around the panel, and then we'll start talking about the burning questions.
Hi, I'm Roger. I'm Roger Packer, and really happy to be part of this symposium, and thank you for the introduction. You know, we're in the beginning of this. We know what we're dealing with, which are diffuse intrinsic pontine gliomas. As Sergio said, we've made no progress in 3 or 4 decades, or ever, where 90% of the kids are dead within 18 months. So I do think we have some opportunity to try to be innovative and take some risks in this population, as long as we do it carefully. And we know for the majority of these patients have non-enhancing tumors. We have drugs that seem to work in laboratories or molecular targeted therapies, and then can't translate that into any improvement in survival. So our initial approach was to work with Incytec. We were able to put in the first freestanding focused ultrasound system, both for HIFU and LIFU, in a pediatric hospital into one of our MRI units, and convinced our institution to allow that unit to essentially go down for 3 or 4 hours while we do this procedure. You know, we have 2 studies that we are now open and accruing patients, and we strongly believe, as per the discussions before, that these can't be institutional studies. They have to be group studies because of the numbers involved. We can try them in one site, but we've got to do the same thing at multiple sites to make any progress. So the first study that opened was a sonolescence study using the 5-ALA to try to activate that in DIPGs. That study is open, accruing. We have treated 2 children. Initially, the FDA, and that's part of the regulatory process with this, it took a long time, allowed us to treat half of the brainstem and then come back and treat the other half of the brainstem in these patients. The first patient we treated was a 5-year-old. The second is an older patient on compassionate use. And now, as that patient has gone through the DLT period of 1 month for the second treatment, we are about to treat that a third patient with the entire brainstem. And selection is very careful in these patients. We're trying to treat them after radiation, before they relapse. So there's a really tight window, and also while they're still at least semi-contained in the brainstem, and we understand all the risks of dissemination and missing the leading edge of these tumors, but we've got to start somewhere. We've seen no toxicity except for hypothermia in the first 2 patients, who may be related to having brainstem gliomas, being in a cool MRI area. That's been no sequelae, but I think you have to realize that these kids are going to be sedated for about 2 to 3 hours to get through the procedure, to place the frame, then to inject the 5-ALA, then to do the treatment itself, which takes an hour or 2, and then to observe them. That study will be expanded to 2 other sites fairly soon, and we hope to be doing that at 3 sites. The second study is one we are doing in collaboration with Dr. Lipsman and Dr. Rutka. It's really their study that they allowed us to be part of, using blood-brain barrier disruption and daunorubicin, based on some of the preclinical data that came out of Toronto as far as daunorubicin, if it gets in, causing cell kill, and the ability to then give that medication after we disrupt the blood-brain barrier with microbubbles. That study is also FDA approved, IRB approved, and we're looking for the first patient to be treated, we hope, in the next month or 2, although we believe our colleagues in Toronto will go first in a collaboration we're doing together. We hope other sites will also be part of this soon. And this is what we're trying to do, is to work together to try to make this happen. We know we may not be using the right agent initially. We know we have things to work out, and Dr. Nazarian will talk about some of the preclinical work he's doing and we're doing in collaboration with Virginia Tech to try to be smarter. We're not trying to reinvent the wheel. We want to take the information that Toronto and other sites have already worked out as we try to figure out what the right dose and schedule of these drugs are. But we know we have to keep pushing ahead. We've made the commitment to do it, and I'm hoping that our next set of studies will even be more complex, possibly adding immunotherapy as a better deliverer with the focused ultrasound to the brainstem, and then ultimately moving from the brainstem to other parts of the brain, especially deep-seated areas of brain that we can't reach. Last thing I have to say, that we take a look at all of these studies in a swollen brainstem as being critical toxicity studies. We know there's going to be cytokine release. We know DNA fragments are going to scatter through the body. We understand all of those risks. We may not know how to control them, but this is part of the deal of treating DIPGs, but I think it's the right thing to do.
Thank you so much, Dipg. Maybe I'll move on to Dr. Saltoni from Columbia University to give us a brief update of the previous study and what the future plans are.
Sure. Thank you. First of all, thank you so much for the opportunity to be here and be on this panel. I'm Dr. Luca Salonta. I am a pediatric neuro-oncologist and the associate lead of the Initiative for Drug Delivery Innovation program at Columbia University, which was initially started by Dr. Sakuroulis. And my partner in crime, a radiation oncologist, is Dr. Cheng-Chia Wu. So we have this unique opportunity to have both a radiation oncologist and a neuro-oncologist to team up and lead the program. We have a very extensive preclinical laboratory led by Dr. Wu, and we are collaborating, as we showed before, with many Columbia-led team, including neuropathologists, computational biologists, immuno-oncologists, and also neurosurgeons with other drug delivery techniques. So we had the opportunity to initially do all the preclinical studies, and we were first able to show that the blood-brain barrier opening in the brainstem, which is a very delicate area of the brain, is safe. And based on that, with a DIPG model, a mouse model, we were able to show that panobinostat, an HDAC inhibitor, which was very promising in the preclinical trials before, we proved that it improved survival in the DIPG model. So initially, that was the basis of our very first pediatric DIPG trial for progressive DIPG to use focused ultrasound with panobinostat oral treatment. As Dr. Zacharoulis has mentioned, we are using Dr. Elisa Konofagos' TheraWave system, which is a little bit different from the Insightec system. The few differences that it can be administered while the patients are awake, so we don't need sedation, and it's not MRI guided. It's using the same navigation system the neurosurgeons use. It's a fairly brief procedure. It takes about 2 to 5 minutes, the whole procedure, with a bolus microbubble infusion or injection, and then followed by a very brief, about 2 to 3 minute focused ultrasound treatment. So that was our first experience in the pediatric field, and we were able to treat 3 patients with this during this study. It included about 25 total treatment sessions, and most of our toxicities only included some skin irritation at the site of the ultrasound. Since it was not powered to look for survival, but it was more like a phase 1 feasibility safety study, we could conclude that the blood-brain barrier opening in DIPG patients was safe and feasible. We were able to incorporate with panabinostat oral treatment using focused ultrasound treatment about every 2 to 4 days. And also what we were able to see that there were 2 patients out of these 3 with symptom improvement. As some of you might know that panabinostat, oral panabinostat, was actually removed from the US market. So we needed to look for other oral drugs to try to reopen and a little bit restructure this study. So our next steps in the pediatric studies that we're trying to use etoposide with the similar structure and method of the study. We are currently, as we speak, we are sending the IND application to the FDA and hoping to reopen this study in about 1 to 2 months. Because we have other computational methods and immuno-oncologists in our team, we are actively looking for other ways to making sure that we're using the right drugs and the right concepts with our focused ultrasound studies. We are looking into drug combinations, not only single drugs, with our computational methods for DIPG, and we're also planning to use checkpoint inhibitors, either CD47 antagonist or PD-1 antagonist to combine with FUS. And those are in preparation for further pediatric clinical trials.
Fantastic, thank you so much. Just to point out that the 2 patients who had the clinical improvement had radiographically stable disease.
That's correct.
Which brings a bit of a question, what was the explanation of the clinical benefit? Perhaps the vascular changes, which is something that we don't know, but quite interesting first data. Thank you. Maybe I'll move on to Kevin Beccaria, if you want to mention the French experience and updates and the status of the future studies.
Good morning, and thank you for the opportunity to be here today and to talk about our experience. Our clinical experience is just beginning. The sonopid clinical trial has not begun yet. We are ready, finally ready to begin, and we are ready to recruit the first patient. We will be recruiting in the next few weeks. The main difference in our trial compared to the other ones is that we use a sonoclot device, and the sonoclot device is a non-focused ultrasound device, so that we will be able with this device to target a large volume of brain, of parenchyma. For this trial, there is one particularity. We decided it's a phase 1 trial, so we just want to check the feasibility and the tolerance of the blood-brain barrier disruption with a sonoclot device in the pediatric population, and we want it to be safe. And we decided to focus in this first trial on the supratentorial tumors, just to be sure there's no issue with the posterior fossa, with big brainstem with edema, and to be sure that we're able to open the BBB in the pediatric population and then to go forward to a second step to the brainstem. We also decided not to focus only on diffuse midline gliomas, and we will include all patients with malignant supratentorial tumors when relapse, just to have enough patients, because we are in very, very small pediatric populations. This trial will be an escalation dose trial. We will begin with 2 kinds of escalation. The first one is the dose, the intensity of the ultrasound, the pressure, the acoustic pressure. We will begin at a small acoustic pressure and increase it until we will be able to target the efficient acoustic pressure, probably we will target and use the acoustic pressure that is used today in the adult population. You know that the sonoclot device is used in the adult population in glioblastoma, and that there are a lot of data on it. So we will just check that the ultrasound, the acoustic pressure will be the same in the pediatric population. And the other aspect is that we will use the sonoclot device 9, the device with 9 devices in it. And since we have never used it in the pediatric population, we decided just to begin by implant the sonoclot 9 device and just activate 3 of the devices of the ultrasound devices for the first patients. And depending on the tolerance of the opening, we will go after that to an activation of all 9 devices, all 9 ultrasound devices for the last patient. So it will be 2 steps of escalation with the acoustic pressure and with the number of devices activated. We decided to treat the patient with carboplatin because carboplatin is known that this drug has been widely used in the brain, and we know that there's no big issue with high dose of carboplatin, and we have lots of data with a sonoclot device in adults, so it was a safe point also to do when we decided to use this drug. So this is our plan, and we are waiting for the first patient to be recruited.
So, and you anticipate the study to start enrolling soon?
We are ready, and we are just waiting. Yes, we are just waiting for the first patient.
Fantastic. Thank you. Thank you so much. And Dr. Van Woerden, Dennis, if you want to give you...
Yeah, great to present. Yeah, so in the Netherlands, we are just starting focused ultrasound in the clinic. So we do have a program preclinically in our hospital in close collaboration with the UMC Utrecht. So that's the Princess Maxima Center. And we closely work together with UMC Utrecht. And my colleague, Dr. Mario Ries, is actually in the audience. So if you want to ask more, you can ask him. So we recently established the Dutch Neuro-Oncology, NeuroFus Consortium, which is not only neuro-oncology, but also researchers interested in Alzheimer's disease and Parkinson's disease to make use of the focused ultrasound platform, which we are currently establishing clinically. And that's an Insight Tech 4000, which is being integrated into a Philips Ingenia scanner at the moment. And we expect to start trials early next year. And one of the first trials we will start is in diffuse midline glioma, so both pontine and thalamic tumors. And we aim to include 10 adults and 10 children, and just on the backbone of Stoop, so with temozolomide, to prove feasibility, because it's one of the first clinical trials in children in Europe. So that will be a first feasibility study upon which we will build later on. One of the other studies will be monoclonal PET-labeled monoclonal antibody for Alzheimer patients, whether that be directed against amyloid or tau, that still needs to be discussed. So, and for the preclinical part, we work in the lab looking mostly at pharmacokinetics, since we realize that pharmacokinetics are really important. We need to align plasma levels to what we want to aim for in the brain, meaning that when you open the blood-brain barrier, drug gets in, but it also gets out if your pharmacokinetics are really quick. Like, for example, the doxorubicin, and you need to investigate whether it's either Cmax or AUC that's important. And of course, most of the drug sensitivity assays in the lab have been built on 72-hour exposures to certain drugs, which we will never get in general in the patient. So I think we also need to mimic what we will do in the patient more in the lab. So that's what we do on microfluidic on chip systems, try to mimic as much as possible the pharmacokinetics in patients. And one other part, Utrecht is really strong in immunotherapy. So what we also want to do is combine immunotherapies with focused ultrasound. So increasing the local immunity, boosting microglia and macrophages, potentially in concert with CAR T-cell therapies in preclinical models and hopefully later on in clinical studies in adults and also children, potentially via the PNOC network that Javad and I'm in. So I think it's really very, very promising, the field, but a lot of investigations need to be done. And we were discussing, Javad and I, before this meeting, that we really need a network of people together so that we collaborate and discuss and harmonize procedures, both preclinically and clinically.
That's very
So,
exciting.
that's
Thank you,
it.
Dennis. And Javad, Dr. Nazarian, if you want to give an overview of the efforts, the initiatives that are being done in the translational research.
Sure. Thank you very much for the opportunity, and thank you very much for the introduction. As Dr. Hacker mentioned, I'm a basic biologist. I work with Roger at Children's National, and also I lead the team in Zurich. We've been fortunate early on to work on some of the sonodynamic therapy aspects of focused ultrasound, some of the work that has been translated to the work that Dr. Packer mentioned. But in the context of clinical trials, as Dennis mentioned, we are trying to develop correlative studies where we can take CSF or plasma from the patients and measure a number of different biomolecules or molecules that were mentioned here today. We have shown the feasibility of collecting and analyzing ctDNA from plasma, but also from CSF of the patient. ctDNA could give us a number of clues. One is the openness of the BBB, with the idea of the longer that the BBB is open, the higher the amount of or quantity of ctDNA that you could potentially detect in the plasma or in CSF. But you could also look at the tumor heterogeneity, which is something that potentially biopsy itself cannot inform us. We're also developing platforms where we're looking at the engagement of the tumor with the immune system. Again, the opening of the BBB would give that opportunity for some of the immune activation potentially. The clinical trials that are being launched now gives us at the preclinical sites the specimen that we can analyze and we can assess and develop these platforms to look at the peptides that are potentially present within the microbiome of the patients, within the plasma or the CSF. But we also understand the paucity of the correct models to analyze this. Some of our colleagues today presented the murine models that we have, which have been great to inform the trials that are opening now. But we also realize that we need to move into larger models, especially if you're talking about the focused ultrasound versus larger opening of the BBB. To that extent, we have been working with Columbia University, our colleague presented here today, and Virginia Tech to form the first DIPG DMG funded working group that has engineers, preclinical folks, and clinical colleagues that are now looking to address some of these questions. We are going to develop in vitro models. This will be 3D cultures at Virginia Tech and Columbia. We'll be developing in vivo models, large and medium and potentially larger size, small and medium and potentially large animal models that we'll be testing the openness of BBB and the effect of the healthy cells as well as the tumor cells themselves. And as Dennis mentioned, now we have formed the first focused group with specifically focusing on DIPG and DMG. But there is a need, and again, as Dennis mentioned, to expand this to the other groups who are interested to bring in their expertise as well.
Thank you so much. We need the whole morning to discuss all the burning questions, but briefly to address some of the controversial issues and to see what is being done across the various trials. So one big question is, what is the imaging modality used in the planned studies in order to determine the blood-brain barrier opening? For example, when we started the study at Columbia, we still use contrast enhancement. But one practical issue that came with this is that sometimes there was uncertainty of to what degree the contrast was avid enough to consider that we have a confirmatory opening. So there is an obvious need for more sophisticated ways to validate the opening, and I'm just curious from Dr. Packer what the plan is in terms of the imaging.
For the sonolescence study, we're not really using blood-brain barrier as a criteria. We're looking at the treatment effect more than anything else, and hopefully the circulating DNA and other things that will give us some parameters. For the blood-brain barrier studies, we're dealing with children that we're sedating, and we can only sedate them so many times during the day. And we can only, and it sounds a little bit crazy because we're dealing with two patients that have just received 5,900 or so CGY to the brainstem, but we have radiation safety parameters of how often we can image them, and we can't really use PET repetitively. So our plans are we image right in the scanner because the InsightX system is in the scanner prior to the initiation of the treatment, and they get another scan at the end of treatment to look at barrier disruption, and a third scan at about 24 hours afterwards. So they will be getting 3 scans with contrast. Right now, the time, we haven't done the first blood-brain barrier patient, but we expect that they're going to be sedated with the frame on, and we will be doing the imaging with the frame on, obviously, for up to 3.5 hours. So that's a long time to keep a kid under. The second scan will be, the last scan will be much more rapid. So we were limited, but we're going to be using MRI as our criteria.
For the 3.5 hours under anesthesia with the potential of having the blood-brain barrier open, do you think there's a concern of neurotoxicity?
Well, I think that, with the, as Dr. Lipsman talked about previously, we're using bolus microbubbles, but it takes time to put the frame on. I mean, we've gone through this process. It takes time to place the frame. That's done, the head is shaved at this point. The frame is placed on the child. The child is asleep during that period of time. Then the child is transported from the room next door to the MRI suite, placed in the MRI suite. The actual sonifications of the treatments may be 2 hours, so they may be less. We're trying to change those parameters, especially on the sonolescence study. But then you're going to do another MRI, so you add it all up. It's about 3.5 hours. The actual treatment with the microbubbles treatment is maybe 2 hours. Then they go to the ICU, and because we think the blood-brain barrier disruption will last somewhere between 6 and 8 hours, they will likely get their infusion in the ICU, although we're still working that out. We may give the infusion right into the recovery area to try to not miss a time window. But then there are 24 hours of observation afterwards on both studies in our ICU. I just have to do a shout out. To do this in a pediatric population is you really need the help of everybody. Anesthesia, radiology, neuroradiology, the PICU, all of those people have to be on board and working with you as a team because our time window is so tight. So we had multiple walkthroughs that Dr. Kilburn and Dr. Fonseca from our group led, working with Dr. Sharma, the focused ultrasound person, and our neuroradiologist and our anesthesiologist to make this happen. So far, they're going well, but you cannot be cavalier about this because the timing is so critical.
Thank you. And Dennis, I know you're planning some much more sophisticated imaging, if you want to elaborate a little bit on the PET scan.
Yeah, and I think it's a good point you just mentioned, Sturgis, that the anesthesia itself might be causing more toxicity when you have the blood-brain barrier open. So we need to closely look at that as well. But yeah, so what we will do is, especially for long-lived molecules such as monoclonals, it's very elegant to label them with zirconium, so you can actually image them over time, but even after a few days, and see whether there's increased uptake after focused ultrasound. So what we will do in Alzheimer's disease is compare a group of patients that did not get ultrasound to a group that do get ultrasound. And for small molecules, it's slightly more difficult because you need a cyclotron at your site, especially for 11 carbon labeled molecules, but for 18 fluoride, you might ship that. But I think PET imaging is also an interesting option to look at whether you have increased drug uptake in patients. And there are even people who think you might integrate a PET MRI with a FUS helmet, and that's maybe very far-fetched and maybe not needed. But I think we need to look better at whether we have better drug uptake only than gadolinium, because gadolinium is quite a big molecule, right? So it's much bigger than our small molecules that we want to deliver. So if you are gadol negative, it might not mean that you're drug negative in your tumor after ultrasound. So I think combining it with PET might be an interesting way, but it's not possible in every center.
I see. And do you incorporate also spectroscopy in combination with the PET scan?
Yes, in general, we do that in all our patients, but so we plan to add that as well in our study, yes.
I see, fantastic. And Kevin, in France, what are you planning to do for imaging?
We plan to use the MRI with a pre-treatment MRI 48 hours before the sonication and a new MRI just after the sonication. We decided for this phase 1 trial to include only patients who will be able to go to the MRI without any sedation, just to not have any issue with the drugs for the sedation. So it will be only patients more than 5 years old, and we know they are able to go to the MRI. And we will use multimodal MRI with gadolinium enhancement, which will be the main point to check the opening of the blood-brain barrier, and some perfusion imaging and arterial speed labeling imaging.
Fantastic. And we don't have a lot of time, but one major issue is biomarker evaluation, given how difficult it is to image the pontine gliomas. Lucia, if you want to mention what you're planning to do.
Yes. So with the upcoming etoposite study we are planning to do, we would like to also collect serum for cell-free DNA. We believe that it's just as important what's coming out after we open the blood-brain barrier as much as what we can deliver to the brain. We are using a CLIA-certified test called MSK Impact, which is looking at about 500 different cancer-related genes and validated in the CSF and the serum for cell-free DNA. So we are planning to get serum samples before the sonication and after the sonication in about 4 to 6 hours based on the half-life of the cell-free DNA to try to predict the tumor response to the treatment, also to see if we can predict the tumor progression sooner than we can see it on the MRI, and also trying to see how etoposide works on the tumor.
And Javad, just because we're out of time, if you want to make a comment of how you coordinate all these efforts for all the trials?
Sure. No, I think for us, this is the most exciting point in the treatment of patients with Dipg post taking an actual biopsy. It's going to produce tremendous amount of specimen that we can work with and ask the right questions. And the group that we have now with Columbia and Virginia Tech and with Dennis and his colleagues will be looking at some of the preliminary work that we have, who has a CLIA certified, who has the most sensitive platform that can be used, say, for ctDNA, and where are the paucities and deficits that we can perhaps fill within our working groups.
Please, yeah.
I just wanted to do a shout out to the FDA. We criticize the FDA for being obstructional or slowing things down and not understanding what we're trying to do for this patient population. We expected the FDA was going to tell us to a 10th of the brainstem and a 20th of the brainstem. The FDA feedback after we gave our first study is that if you're going to treat these patients, treat the entire brainstem. So they've told us that every patient in the study ultimately has to have the entire brainstem treated. For the senescent study, the first patient at each level gets 50% and comes back in a month for the other 50%. The blood-brain barrier study, patients get treated over 3 months, first 50% and 75% in the third month, 100%. So all patients on these studies will get the entire brainstem covered. And this was shocking to us from the FDA. We thought we would get tremendous pushback because the major side effect is, of course, we could kill the child if there is too much cytokine release or too much swelling. So we appreciate the FDA's support in this, trying to make a meaningful difference for these patients.
I will echo these comments with the FDA experience. We were very surprised, positively surprised with their will to support such innovative approaches for this horrible disease. So yeah, we're very grateful. Maybe a last comment from each team of the panel, primarily in terms of their future outlook, in terms of safety, and what are the potential agents and the drugs of choice that they would like to see be tested over the next few years after these first initial safety studies are completed. Maybe I'll ask from Lucia this time.
So we believe one of the ways we can improve safety is we're also monitoring the closure time of the blood-brain barrier. We noticed, based on our experience in our first study, that there can be differences between closure time, and since our study is structured as we are using every other day focused ultrasound treatment, we wanted to make sure that opening the blood-brain barrier only happens after we can prove the closure of the blood-brain barrier. Based on these results, we are right now with the topocyte study, we are structuring our study based on determining the closure time for each patient, and then that's how it's going to determine the frequency of the focused ultrasound treatments. Also, sometimes there's a question regarding spreading of the disease if we open up the blood-brain barrier. And one answer for the future can be using radiation before or concomitantly with focused ultrasound. We already have our preclinical studies proven that focused ultrasound and radiation together or subsequently can be safe. So maybe that can be another way to try to maximize the safety of these patients, knowing the biology of the tumors and expanding from Dipg to other type of pediatric brain tumors. In terms of our future study, as I mentioned, we're hoping to open the topocyte study within a couple of months once we get all the approvals and regulatory documents. We're also working with our computational biologists to try to come up with combinations of therapies which can target different subpopulations of the tumor. And also from the immuno-oncology standpoint, our goal is to combine focused ultrasound with different checkpoint inhibitors, which we were also able to prove preclinically that it improves survival.
Thank you, Lucia. And Kevin, what is your future plan beyond carboplatin?
Well, there are different issues. You discussed the kinetics, and for the first patients, we decided to do the carboplatin injection after the sonication, just for an issue, organizational issue, because we will have to do the MRI just to check the feasibility of the BB opening, and we plan after this first step to go to an injection of the carboplatin just before the sonication, because our first results in adults with a circular device let us know that and let us think that the opening of the BB is largest during the sonication and just after the few minutes after the sonication. In terms of drugs, we are working in the lab, and it will be submitted probably in a few months. But we are working on some kind of radiosensitizers that could be used with the ultrasound and concomitantly with the irradiation.
And Dennis, you had mentioned before some strategies to evaluate pharmacodynamic effects. In terms of the future, how are you planning to implement this in clinical trials and decide about the best drug choices based on that?
Yeah, so I think we need neuropharmacologists to be heavily involved to predict brain PK and AUC. And I think the future, because DIPG and DMGs are still diffuse gliomas, right? So they're not localized diseases. The cells migrate all over the brain eventually. So I think we still need a therapy that covers also. I really believe in the combination of immunotherapy plus focused ultrasound to also with an abscopal effect treat cells that are way more distant than the field you open with your ultrasound.
And Dr. Parker, in terms of beyond the
Yeah, we're going to do future therapy, whatever Dr. Nazarian tells us to do in his laboratory. But seriously, I think the future is going to be combination therapies. We are not going to cure this disease with one drug, with one treatment. We're going to have to figure out a way to do this multiple times safely. We hope the technique will move to a stereotactic frame so we can do them more quickly and more repetitively. It's a great technique, but there are things that have to be worked out. At this point, our study criteria does not require biopsy for entry, but for the next generation it will, because we'll be into either molecular targeted therapy or immunotherapy, and we'll try to figure out what to do. And our hope and our stated goal and our vision is that we will be doing something with immunotherapy, maybe as a single agent, maybe in combination with the hope of CAR T, to be very honest, within the next 2 to 3 years. That's our goal, but we've got to get through the first steps first. As far as your question about safety, I think patient selection becomes extraordinarily important. I know the DIPG community wants us to treat every child, especially those that have relapsed. We just can't do that yet. We're going to hurt someone. We're going to close this program down. We want to have patients who are relatively stable when they enter treatment that could qualify for the study because we're hitting the right target. And I think that's where our focus has to be for patient safety, selecting patients that we think can benefit from this, but also do that without very severe side effects.
Thank you. And Javad, the last comment is on you about using modern technologies to predict how we personalize therapy for DMGs without interventions, without biopsies.
No, I just want to thank Dr. Packer for giving me the world pressure. I will go back to the lab and make sure that we design the next experiments. But again, I really think this is a fantastic opportunity now in the treatment of children with DIPG. We've had a number of HDAC inhibitors that have been really promising in vitro and also in vivo in the laboratory. But we think they haven't made any treatment, any effect in the clinic because they don't get they don't cross the blood-brain barrier. Fimipinostat is a very good drug. Again, it keeps coming up as our #1 drug, but again, we think it's not effective because it doesn't get in. We have seen clinical efficacy of ONC201 for a number of patients, thalamic, but maybe not Pontine patients, but maybe that's also a blood-brain barrier penetration issue. Combination of the drugs where ONC201, for example, that's effective for Pontine combining with an HDAC, fimipinostat, for example, that could potentially have a higher penetration to the tumor bed with the focused ultrasound. I think all of those would be very exciting, but we work very closely with our clinical colleagues to pick our top combination strategies.
Fantastic. Thank you so much, all the panelists, for an incredible session. Again, there are so many other questions that we wish we could address, but we just don't have enough time. But this has been exciting. Thank you so much.
Thank you to Dr. Zacharoulis, and that was an excellent panel. Really gets us motivated for the future and all of the work we have ahead of us. I did want to just put a plug in for the FDA town hall that will actually happen today. So thank you for showing your support and appreciation of the FDA. I also totally echo the sentiments of Dr. Zacharoulis and Packer on how helpful the FDA can be for us. So I really encourage you all to join us for that session this afternoon. We will now take a short break. We'll return here at 10:15 for a really great keynote address from Bob Smith, who is the Senior Vice President of Global Gene Therapy Business at Pfizer. So that will begin promptly at 10:15. Thank you all so much.